afternoon. I'm by name of Adamun Jibrin Chere, a student of biomedical science, level 3. I would like to present about my topic, which is about question from 28 to 30. What is immunocytochemistry? Then, immunocytochemistry, first of all, come with from two words, which is immuno has something to do with antibodies, and cytochemistry, which is deal with antigen-antibody reaction. Then there are several steps to how to performing immunocytochemistry. We have like fixation, permeabilization, blocking, and primary antibody incubation, secondary antibody recreation, detection. Then how is that steps performing? So we are going to show you. First of all, is like a fixation. We are going to fix the cell in the slide or something in order to preserve their structure and immobilize their cell protein. Then in permeabilization, we know that each cell has like permeability in order to prevent the others foreigner to enter to the cell. So then we are doing this permeabilization to allow antibodies to penetrate the cell membrane like that. So after we have our blocking. So in blocking agent like are used to prevent non-specific binding of antibodies to others cellular component. Then like after we are going to add the primary antibody. This primary antibody is going to target the epitope. So epitope, those epitope. So to bind with epitope. After when binding with epitope, we are going to wash it. So during washing, so those like others antibodies who didn't bind with the other cells, they are going to remove out. During like binding with the other antigen, they are going to remove out. Then after it will come secondary antibody incubation. In secondary antibody incubation, it's come to bind to the primary antibody in FC region. So you know that primary antibody has FC region. So that secondary antibody it will come to add with secondary with primary incubation. Then after we come to the detection. In detection, like we have two methods. So the first method is like fluorescence microscopy. In fluorescence microscopy, like deal with a microscope, like to visualize under fluorescence microscope. And the second one is like colorimetric detection. In colorimetric detection, like we are using enzyme linking antibodies, so to change the color. Then the second question is about like what does indirect fluorescent antibody testing work? So which is like IFA. So this test can be used to detect specific antibodies against various etiological agents present in patient serum. So how is it performed? So in this, like first of all, we need a glass. So in order to smear it, so we need the glass to smear it very well. Secondly, like we need a patient serum. So we need a patient serum is to add it on this smear, and after like we are going to what you call to bring the antibody in order to make it like a antibody complex form, antigen antibody complex. So then after we are going to wash the serum in order like to remove out those antibody which didn't bind with the antigen, and after it will come like the what do you call it will come after it will come the secondary antibody. So these secondary antibodies come to bind to the primary antibody. So that's why it can make the difference between direct and indirect. Direct, which is like we need directly the secondary antibody come directly and bind to the antigen. But for indirect, it's going to do indirectly, it needs first like primary antibody and secondary antibody. Then for the last question, it's about what is flow cytometry and how is it used in immunology. Then flow cytometry, first of all, is a powerful laboratory technique used to analyze the physical and chemical characteristics of cell or particle as they flow through a fluidic system. Then the main important question of this case is like how is it used in immunology? So how flow cytometry used in immunology? So, like as you see the what do you call the image? In this image, it's like first of all the sample is prepared by staining the cells. So with like specific antibodies conjugated. So when you say that antibody conjugated is like what do you call it's like antibody which is linked with the enzyme. So this antibody target like cell surface marker that are unique to different immune cell populations. So then the fluorocarb emit fluorescence signals. As you see, those like in this those red like those are signal when excited by the laser, yeah, allowing for detection and quantification of the label itself. So like and we are going to prepare the sample by introducing into the flow cytometer. So like where the cell are hydrodynamically focused in the signal in the single field stream. So as the cell like pass through the laser beam, so they scatter the light in different direction. For one scatter, measure the size of the cells. Why side scatter provides information about the internal complexity or granularity of the cell. So by that, so I am done with my presentation. Then thank you for your following.